an almost done PhD candidate in my lab. Yuseung has been here since 2019 and came as part of this interdisciplinary cohort of students that was interested in studying Greenland, studying the changing Arctic, changing hydrology of a changing Arctic. And so when he arrived as part of this Water Institute Graduate Fellows cohort, we started preparing to think about Greenland under the guidance of the Martins and other faculty, and then COVID hit. And all the plans that we had went out the window. And Yuseung improvised, and he came up with two, which I think are sort of really seminal papers about metabolism in flowing waters that came from the necessity of having to make something out of COVID. And so his first two chapters of his dissertation, which he's going to present today, were born of that. And I think that sort of resilience and improvisation and is really a testament to his character. He came from Korea, has a master's degree from Korea, and I think has embraced a lot about being an American scientist. I know that he's ready to graduate because he's willing to tell me when I'm wrong now, which is very well, it's often, it happens a lot, and now he points it out, which is really, really good. So he's been one of the best students I've ever worked with. He's been enormously productive and creative, but also very generous with his time with other students. His diligence working in Greenland, which sounds exotic and fun, but it's really a mosquito hellhole. It's actually not. It's a beautiful place, but the fieldwork conditions are really challenging. He spent 11 weeks doing fieldwork last summer and 7 weeks doing fieldwork this summer. So he really threw himself into this enterprise in a way that I applaud and I'm grateful for. And we're trying to delay his graduation as much as possible so he doesn't leave us. With that, I'm gonna let him take it away, and thank you.
Yeah, thank you, Matt. Well, that's the evaluation of myself better than I can make for myself, I guess. So hello, everyone. Thanks for coming here. As introduced, my name is Yuseung Shin, and today I'll present about my dissertation work. Okay, so before going into the very details of my dissertation, I want to first briefly introduce about my research field. So I'm interested in river ecosystems, so I've conducted multiple field studies in Florida Springs and Greenland streams. So, and along with those laborious field work, I also work with the sensors, deploy them on the streams so that I can monitor the changes of stream ecosystem, which accompany with some computer work. And for data analysis, I worked on the R software, making thousands of lines there. With this diverse method, my work is based on ecosystem ecology in river with the goal to understand the nature and function of river ecosystem and the link between river ecosystem and surrounding environment. And for doing all this work, there are many people who helped me, and I want to thank to all of them. I first want to thank to my committee member, Dr. Matt Cohen, Dr. Jim Jowett, Dr. John Martin, and Dr. AJ Raysinger for supporting me and giving many good feedbacks. And I also thank to my lab members who always spend most of my time on campus and off campus, and also to Water Institute graduate fellowship crews who have worked together about Greenland fieldwork throughout my PhD. And I list the name here because it's hard to name them one by one. So thank you all for helping me staying in the UF and working my PhD. I also thanks to UF SNRE and FFSG, and I spend special thanks to Karen and the Christy, although she's not here, because they made my academic life in UF very easy. And I also thanks to UF Water Institute and SF for supporting funding my research. So let's jump into the difficult, challenging topics of river metabolism. The topic of my dissertation is the temporal structure and nutrient controls of river metabolism. To begin with, we first need to learn what river metabolism is. In definition, metabolism is the chemical processes sustaining life of living organisms. When we expand the scale into ecosystem, metabolism refers to chemical processes maintaining ecosystem. And there is one principal law of nature saying that the energy input of the ecosystem should equal to the energy output. Generally, the input is the sunlight and the output is heat. There are myriad of reactions that engage in these processes, but there are two most important reactions that we generally refer as ecosystem metabolism. And those are photosynthesis, which convert sunlight into biomass, and respiration, which convert biomass into heat. These rates are quantified as a gross primary production, shortly GPP, and ecosystem respiration, shortly ER. But since it is very difficult to measure the rate and the amount, conversion amount from energy to biomass and biomass to energy, these rates are often estimated by the exchange rate of gases because photosynthesis and respiration engage in the production and consumption of oxygen and carbon dioxide. In reverse, we calculate GPP and ER based on the diurnal variation of dissolved oxygen. We deploy sensor in the stream, capture the diurnal variation of dissolved oxygen, and convert that into rate of DO change over time, which is again decomposed into GPP, ER, and gas exchange. Since GPP and ER is the important terms throughout my presentation, so please remember that GPP is the ecosystem level of photosynthesis, and ER refers to the ecosystem level respiration. To further understand river metabolism, we first need to understand what drives the metabolism. So in this slide, I first brought three main drivers of the terrestrial ecosystem metabolism. First, light drives photosynthesis. Second, high temperature stimulate metabolism, cold temperature constrain metabolism. And high nutrient also stimulate metabolism. Therefore, water availability is also one of the main drivers of terrestrial ecosystem metabolism, but I did not include that here because my focus in the river ecosystem where water is never depleted. Since these three drivers vary together seasonally, we also expect seasonality from metabolism. So we expect high GPP and ER in summer and low GPP and ER in winter. However, that's not the case for river ecosystem because of two main reasons. First, light is strongly attenuated by canopy from open air to river surface and by water column from a river surface to benthic. And because of this strong light attenuation, light availability in river ecosystem is affected by not only by season, but also by network position and ecoregion. And therefore, variation of the light in river ecosystem is more dynamic than just simple seasonality. Next, the flow variability also affects the metabolism, the variation of river metabolism. High flow variability causes benthic scouring, meaning removal of biomass and organic matter on the river bottom, and also cause high water column light attenuation. Therefore, we expect more dynamic river metabolism with more dynamic flow. This paper, published by Bernard and et al. In 2022, clearly state that these two factors, light and flow, regulate the river metabolism. Because of these two ultimate constraints, river metabolism became extremely dynamic, and therefore we did not yet clearly find the universal predictor of river metabolism. And also, since these two factors override the effect of nutrients, we did not yet clearly demonstrate the effect of nutrients on river metabolism. Based on these research questions, based on this knowledge gap, I came up with three research questions, which became the foundation of my dissertation. First, does river share common temporal patterns and drivers of metabolism, even under very contrasting environmental conditions? Second, how important are nutrients for river metabolism across sites? And third, do nutrients drive river metabolism when light is not limited? So let's go on to my first chapter, temporal structure and control of river metabolism. And I thanks to Dr. Chavez for collaborating on this work. To talk about this topic, we first need to know about the concept called metabolic regime. Metabolic regime refers to the characteristic temporal patterns of ecosystem metabolism. This concept describes dynamic variation of metabolism across timescales, for example, from daily to multi-annual. This concept was introduced because river metabolism vary more dynamically than simple seasonality. However, since this concept requires high-resolution multi-annual data, this has been the challenge to study metabolic regime for decades. But thankfully, there was a lots of advancements of DO sensor technology. So nowadays we can collect river metabolism in high resolution with long duration. And with this advancement, we can even deploy DO sensor on the ice sheet in Greenland. And this technology advancements lead to the expansion of high quality long-term data of river metabolism. This paper published in 2018 by Epling et al. Report the river metabolism data across 356 rivers in the US, which enable continental scale analysis of river metabolism. And this Stream Pulse project made the data repository where scholars around the world can upload their river metabolism data. These large data sets stimulate active research on river metabolism, enabling categorization of river productivity regime, making spatial model of river metabolism, and river network simulation of river productivity. However, despite the great insights from this paper, they only focused on cross-site variation using statistics, for example, like annual mean or the indicator of seasonality, which cannot represent the dynamic variation across time scale, especially in intra-annual scale. And therefore, this paper neglected the embedded temporal structures and coupling with drivers across various time scales. And this limitation necessitates the novel descriptive tool for studying river metabolic regime. As that descriptive method, I chose frequency domain. Frequency domain is defined as the signal with respect to frequency instead of time. Since it is hard to understand what frequency domain is just based on the definition, I brought one example how we can convert time series into frequency domain. Time series can be decomposed into the signals composing that time series. This example time series is majorly consisted of three periodic cycles. You can then estimate the magnitude of each periodic cycle and plot them on the frequency domain. Here, the magnitude of each periodic cycle is named as power spectral density, shortly PSD. In the example frequency domain, you can see that there are 3 peaks of PSD which correspond to each periodic cycle found from decomposition of time series. Therefore, frequency domain is an effective way to find uncharacteristic periodicity from noisy time series. So what insights can we get from frequency domain? First, like I mentioned, the characteristic periodicity. And since we expect seasonality from metabolism, I focused on annual frequency. To quantify this seasonality, I came with a metric called relative strength of seasonality, shortly RSS, which is the ratio between the PSD at annual frequency and the mean PSD of intra-annual frequency, which is marked by the blue dashed line on the figure. When you have high RSS, it means strong seasonality, and when you have low RSS, it means weak seasonality. The second property is called fractal scaling, in other words, 1/f noise. In nature, this power spectral density often inversely related with frequency f with the varying slope named as alpha. In this study, I focused on the spectral slope of 1 because it has more implication than the flatter or steeper slope. When this spectral slope is 0, which is named as a white noise, it indicates random variation, and there is no memory of previous time steps. Therefore, this signal is only consisted of a step-to-step variation. When the spectral slope alpha is equal to 2, which we call as a brown noise, this indicates that the signal is the random walk variation, meaning it has the memory of only one previous time step. Since this signal is highly autocorrelated, this signal is consisted of only long-term trend, not by short-term variation. In contrast, when the spectral slope is equal to 1, it indicates that the variation is systematic and implying that there is underlying control. And this signal contains memory of all time scales. And therefore, the signal is consisted of variation of both short-term and long-term. This figure from Hawley 1996 again emphasized that the relative importance of the unit frequency is equal across frequency when the spectral slope is equal to 1. And this paper stated that the pink noise, in other words, 1 over F noise, should regard as the null model for environmental fluctuation. Following this, I wanted to check whether river metabolism also follow 1 over F noise. The last insight we can get from frequency domain is the coupling with drivers. Here, I brought one example of metabolism and 3 frequency domain example of driver with varying slope. When we get the difference between these two, we can get the function called transfer function, which implies a variability transfer from the driver to metabolism. So this transfer function indicates how the variability of driver to the metabolism is amplified or damped, or whether there is no change at all. And among these 3 changes, I focused on transfer function slope of 0 because this indicate that the variation of metabolism and driver aligns perfectly. So using the frequency domain, I first evaluated a temporal structure of river metabolism by converting time series into frequency domain. And by using transfer function, I test the hypothesis that the variation of energy input is imprinted on the metabolic variation. For that, I compared the transfer function of the potential drivers. For GPP, there are 4 drivers: temperature, discharge, surface light, shortly PARS, and the benthic light, shortly PARB. Here, PAR is the acronym of photosynthetically available radiation, which indicates light intensity. And for ER, there are 3 potential drivers: temperature, discharge, and GPP. I obtained river metabolism data from Epling et al. 2018 paper and obtained light intensity data from Savoy and Harvey 2019 paper. Based on the overlap between these 2 data sets and the data quality, the total study sites became 84. This figure shows the frequency domain of all components I studied. From the top, it shows frequency domain of GPP, ER, surface light, benthic light, temperature, and discharge. In this figure, you can easily find that there is a peak of PSD at the annual frequency. And both GPP and ER, the scaling patterns are very close to 1 over F noise, which is marked by the dashed line. However, when you look at the frequency domain of the drivers, you can find that there's a visually obvious slope break at the frequency around 0.1 per day. Therefore, from the frequency domain, I got 3 attributes. First, relative strength of seasonality. Second, spectral slope at intermediate frequency, the frequency between 0.01 to 0.1 per day, and the spectral slope at the high frequency, the frequency higher than 0.1 per day. The violin plot on the right shows the distribution of these 3 attributes. From this distribution, we can find that across river, both GPP and ER shares the dominant annual cycle, which is indicated by the RSS around 2 to 3. This RSS of 2 to 3 means that the seasonality is 2 to 3 orders of magnitude higher than the intra-annual variability. We also find that the spectral slope is close to 1, indicating that the GPP and ER exhibit near 1 over F noise. Since these attributes are quite well constrained in the normal distribution, we can say that these 2 patterns are the core structures of river metabolic regime. And when we compare these attributes between metabolism and drivers, we can find that the attributes of benthic light is the most similar to GPP, and the attributes of GPP is the most similar to ER. To test this relationship between metabolism and driver, I examined the slope of transfer function. And as I mentioned in the method part, I try to find the transfer function slope of 0. Among the drivers, I found that the benthic light has the best variance alignment among all drivers with GPP, and the GPP has the best variance alignment with variance alignment with ER across frequency ranges. Therefore, I concluded that the variance of energy input, which are benthic light and GPP, is imprinted on the variation of metabolism. To conclude, first, I found that there's a universal temporal pattern of river metabolism, which are dominant seasonality and second, a fractal scaling. And I also found that there's alignment of variance between energy inputs and metabolism, specifically benthic light for productivity and autotropic carbon supply for respiration. Since metabolic regime is the concept of temporal variation, I anticipate that using frequency domain on river metabolism can provide benchmarking properties of temporal patterns to better understand and predict river metabolism. The next topic is the nutrient limitation of riverine growth primary production. To begin with, I'll first introduce the relationship between the productivity and nutrient supply. We generally expect higher productivity with higher nutrient supply. However, when there's other limiting factors for river ecosystem, mostly light limitation, the increase of nutrient supply does not lead to increase of productivity. And at certain level of nutrient supply, when the system lies on that diagonal line, we call that system nutrient limitation. And if the system lies on that dashed line, we call that system nutrient saturation. Since river ecosystem is extremely diverse and share myriad of limiting factors, I wanted to know first how common nutrient limitation is in river ecosystem. And second, if the river is limited with nutrient, how limited the nutrient is, and if the system is saturated, how saturated nutrients are in the system. To answer those questions, I use the metric called autotropic uptake length, shortly SWA. SWA is the indicator of nutrient limitation in terms of productivity. We can calculate SWA as the balance of nutrient supply and demand. Nutrient supply here is the multiplication of discharge and nutrient concentration, and the demand is the multiplication of stream width and autotropic uptake rate, the value derived from GPP. When you have low supply and high GPP, we expect a short SWA, and when you have high supply and low GPP, we expect SWA to get longer. This metric has been used to examine nutrient limitation, and King et al. 2014 paper found that nutrient limitation is more likely when this SWA metric is shorter than 10,000 meters. Following this, we use SWA of 10,000 meters as the threshold of nutrient limitation and saturation. The one thing to be cautious here is that this threshold is not a definite standard, but just suggests the likelihood of nutrient limitation and saturation. Using this metric, I developed 3 hypotheses. For that, I calculated autotropic uptake length for 2 major nutrients, nitrogen and phosphorus. The first hypothesis is that nutrient limitation of GPP is rare in flowing water, saying that the SWA of the most rivers will be longer than 10,000 meters. Why? Because most rivers experience light limitation. This light limitation causes low GPP, which ultimately lead to low nutrient demand, and which again cause a long SWA. Therefore, we expect most river sites will be located in the no limitation zone, in other words, far above the threshold. The second hypothesis is that far shortens in summer due to the variation of GPP and flow. A GPP and flow has the seasonality. GPP is high in summer and discharge is low in summer, which means in summer we expect higher nutrient demand and lower nutrient supply. When we combine this, they lead to a short flaw. Following this variation, I expect flaw to get short in summer and long in winter. Last hypothesis is that flaw lengthens with increased watershed area. This is due to river network effect on nutrient supply and demand. Both supply and demand is affected by watershed area, but normally the nutrient supply changes faster than demand. And therefore, in large watershed, we can expect a longer flaw, and in contrast, in small watershed, we expect a shorter flaw. I collected nutrient data from National Water Quality Portal, discharge and width data from USGS, and river GPP data from, again, Epling et al. 2018 paper. From this data set, I was able to collect total 6,135 graph samples from 137 rivers in US. Using this data, I calculated autotrophic uptake length of all graph samples and then calculated annual mean flaw, which can represent the overall nutrient status of individual rivers. This figure shows the distribution of autotrophic uptake length of nitrogen and phosphorus. You can easily observe that many of the points are located in the right top section, which indicates a no limitation. Oh, and before that, I should let you know that the gray dot indicates the autotrophic uptake length of 6,135 graph samples, and the black dots indicate the annual mean autotrophic uptake length from each river. When we convert their frequency into percentage, we can find that around 60% of the sites is likely to be not limited with both nutrients. In other words, around 40% of the sites is likely to be nutrient limited. And when you compare nitrogen and phosphorus, you can find that the frequency of phosphorus limitation is way higher than nitrogen limitation. And that's also shown by the general difference between SWAN and FWAP. The SWAN is generally longer than FWAP, means that the nitrogen limitation is less likely than phosphorus limitation. Next, about the temporal variation of autotrophic uptake length. This figure shows the monthly variation of autotrophic uptake length. The line plot shows the monthly variation of SWA of each river, and the violin plot shows the distribution of all graph samples at each month. You can find that both violin and line plot shows the seasonality, decline from winter to late summer, and incline from summer to winter. Therefore, we can say that there's a general seasonality of autotrophic uptake length, showing shortest value in July to September and longest value in December to October. This pattern is caused by both GPP and discharge, as I hypothesized. The short flaw in summer matches with high GPP and low discharge, and the long autotrophic uptake length matches with low GPP and relatively high discharge. Last, about river network effect. This figure shows the changes of nutrient supply, the brown dots, nutrient demand, the green dots, and the autotrophic uptake length, the black dot, with the changes of watershed area. When you see the changes of autotrophic uptake length, you can easily find that this flaw is strongly positively related with the watershed area, which says that there is a strong area effect on autotrophic uptake length. In other words, nutrient limitation status. This variation is caused by faster increase of nutrient supply than demand. As you can see the difference between the brown and green dots. Therefore, we can say that the nutrient supply is more important determinant of the autotrophic uptake length. In summary, we first found that nutrient is not a universal driver, but can be potentially important driver for river productivity because there are around 40% of the study sites that shows high likelihood of nutrient limitation. And when you compare two major nutrients, phosphorus limitation was far more likely than nitrogen limitation. And second, we found that nutrient limitation is more likely in late summer when GPP is high and discharge is low, and in small streams where the nutrient supply per benthic area is low. This research has implication of river management. Nowadays, we expect a higher nutrient loading from climate change and land use change. And under this circumstance, this research can contribute to river metabolism by guiding when and where rivers are potentially more affected by the increase of nutrient loading. Yeah, I need to hydrate myself. Okay, the last chapter, the nutrient limitation in Greenland streams. You can see that there are like many collaborators in this research, and I want to thank all of them for making this research successful. Before going on to the main context, I want to remind about the relationship between the productivity and nutrient supply. Under nutrient limitation, higher nutrient supply leads to higher productivity. Therefore, when there's addition of nutrient, general expectation is also higher productivity. However, this relationship can differ with the varying environmental conditions. Under dense canopy cover, which is a common condition in temperate and tropical regions, the addition of nutrients does not lead to increase of productivity because light limitation is constraining productivity there. With less canopy cover, we can expect higher productivity response to nutrient addition. However, there's a likelihood of secondary nutrient limitation which constrain productivity. And under this situation, the productivity response to nutrient addition can be lower than the expectation. In the Arctic, where we expect no light limitation because there is no canopy, and where we expect high likelihood of secondary nutrient limitation, we also expect extremely low nutrient supply, which constrain the initial level of productivity. And because the initial level of productivity is also very low, the nutrient addition can lead to high response of productivity, even with the secondary nutrient limitation. However, across Greenland streams, this secondary nutrient limitation can differ spatially. This map shows 5 study sites of my experiment, 3 in coastal area, 2 in inland area. 3 coastal watershed, which is located nearby the town called Sisimiut, they experience longer exposure from ice sheet after glacier retreat, which happened around like 10,000 years ago. And therefore, this site is likely to have very depleted level of geogenic source nutrients. In contrast, Lake Helen, the inland watershed, is located much closer to the ice sheet and therefore experience relatively a shorter exposure from ice sheet. And therefore, compared to coastal stream, it is likely that geogenic source nutrient is less depleted in this area. This spatial heterogeneity can cause the different level of secondary nutrient limitation and also the different productivity response to nutrient addition. Based on these 3 conditions, I developed 3 hypotheses. First, nutrient limitation is prevalent in Greenland streams. Second, nutrient from weathering is more limited at coastal than inland streams. And lastly, higher productivity is expected with a consistent elevation of nutrient level. For hypothesis 1 and 2, I conducted experiment called a nutrient diffusing substrate, shortly NDS. NDF is the cup with agar solution inside and the treated glass on top of that. Agar solution support microbial growth, and treated glass is used as the substrate where biomass can accumulate. The benefit of this method is that you can amend nutrients inside the agar solution. In this research, I amended 3 nutrients: nitrogen, phosphorus, and iron. This photo shows the NDF set deployed in streams. After around 2 weeks, there's a microbial growth on each NDF, and we retrieve them to compare microbial growth between the control and treatment. For the comparison, I measure chlorophyll A on each NDF. Here, chlorophyll A indicates the amount of autotrophic biomass. And after that, I calculate the response ratio, which is the ratio between chlorophyll A on treatment and chlorophyll A on control. If nutrient actually cause the increase of productivity, then chlorophyll A on treatment should be higher than chlorophyll A on the control, which means that we expect response ratio to be higher than 1 under nutrient limitation. I also conduct two-way ANOVA following the method described in Tank and Dot 2003 to examine the co-limitation of multiple nutrients. I skipped the details of this method, but this method is used to separate the primary and secondary limiting nutrient, as you can see in the example box plot. This map again shows my study sites, and we conducted an NDF experiment twice in the summer of 2022 to check the consistency of the nutrient limitation across season. This figure and table shows the result of NDF. The box plot shows the response ratio, and the table summarizes the types of nutrient limitation we found from each site in each season. Since this figure and table contains lots of information, I'll point out the main findings one by one on the following slides. And for visual conciseness, I removed the response ratio lower than 1 because we are only focused on response ratio higher than 1 to detect nutrient limitation. Fourth, about the prevalence of nutrient limitation. I put the red scale about the nitrogen treatment, and we can find that the nitrogen limitation was prevalently found across Greenland streams. So we can conclude that the nitrogen limitation is a prevalent nutrient limitation in Greenland. The reason for this is the extremely low nitrogen availability in Greenland streams. The line plot here shows the temporal variation of nitrogen availability, and the highest level we found from all sites is around 0.08 milligram per liter, which is order of magnitude lower than the mean nitrogen availability in US River, which is around 1 milligram per liter. So we can conclude that this low availability of nitrogen leads to cause the importance of nitrogen in constraining productivity in Greenland streams. Second, about weathering nutrients. We found phosphorus limitation from LH6 and all 3 coastal streams. Just based on frequency, we can say that phosphorus is more important in coastal streams than inland streams. And when we compare the role of phosphorus in each stream, phosphorus was secondary limiting nutrient in LH6, while it was one of the main limiting nutrient in coastal area. Therefore, we can say that this weathering nutrient phosphorus is more important in coastal area. This story is also similar to iron, but iron shows more distinctive difference between the coastal and inland. In inland streams, we were not able to observe any iron limitation, while iron limitation in coastal area was very, very strong. This difference is caused by the different iron availability between 2 areas. As you hypothesized, iron was more depleted in coastal streams, which caused higher importance of iron in these areas. The final piece of my presentation today, the hypothesis 3, which is about the higher productivity with consistent elevation of nutrient level. I conducted NDS experiment with continuous nutrient dosing to test this hypothesis. In a stream, I installed a 2 nutrient pumping station which persistently enrich nitrogen and phosphorus. And this pumping divide the stream into three reaches: control, nitrogen enriched, and nitrogen phosphorus enriched. Here we deployed the NDS to compare the background biomass and response ratio among the reaches. Due to limited effort and time we have, we picked one of the 5 sites, which is the LA6, where we found a distinctive nitrogen limitation and co-limitation of nitrogen and phosphorus. This is the map of LA6 where we deployed a pumping station, as you can see in the photos. And then we deployed NDS, and the experiment was continued between June 25th to July 15th of the summer this year, with the help of the Sam, my lab mate, and my advisor Matt. These 2 box plots are the results of the experiment. The top box plot shows the chlorophyll A of the control NDS, which is the background biomass of the system. And the bottom box plot shows the response ratio. With nutrient dosing or nutrient pumping, we found that background biomass increased. With the nitrogen addition, that background biomass increased, and with the phosphorus pumping, that background biomass dramatically increased. And we also found that the nutrient pumping led to the decrease of response ratio, as you can see in the bottom box plot. These results clearly show that the consistent elevation of nutrient level did stimulate productivity and reduce nutrient limitation of this system. As a conclusion, I answered to each hypothesis in a very short way. First, yes, we found prevalent nitrogen limitation. And second, yes, a weathering nutrient was more limited in the coastal than inland stream, especially for iron. And third, yes, we found a clear effect of nutrient pumping on the stream productivity. This research has implications on the Arctic streams because the Arctic is experiencing the fastest warming in the globe, which causes higher nutrient export by the permafrost melting. Based on our research, we can expect that this higher nutrient export can lead to higher productivity of the Arctic streams. However, our study also suggests that this productivity ratio can vary with the distance from the ice sheet and also by the consistency of nutrient level elevation. Therefore, further studies on the Arctic ecosystem should consider the nutrient effect on the stream ecosystem with the varying environmental conditions. As a takeaway, I made 1-sentence takeaway for each chapter. First, energy input imprint seasonality and fractal structure on river metabolic regimes. Second, nutrient limitation of river productivity is more likely during summer and in small watershed. And third, in Arctic streams, nutrient level elevation causes higher productivity. This is the end of my today's seminar, and thank you all for coming here and listening to my presentation.
Mic to work properly. There we go. Thank you. That was very interesting, and I appreciate that was very informational and educational as well. So we have some minutes here for questions, if anybody has any questions.
So in like your second chapter, you talk about the nitrogen uptake length being longer than phosphorus, right?
Yeah, right.
So how does that, and also in the Leland line, say nitrogen, eliminate phosphorus, less, particularly in coastal watersheds. So how do those results square with it, or how does the increase in nitrogen in those ecosystems as human activity that they would buy processing stuff. Affect your results. Are these all recent, or do you think these have changed since the amount of reactive nitrogen has increased in Earth's ecosystem? That's number one question. And the number two is, particularly in Greenland, the coastal watersheds that were older and more exposed, do you think that there's been more weathering of the apatite mineral? That's, I assume, the dominant phosphorus source there. So do you think that's been largely removed, that makes that more limited there as opposed to?
Yeah, for like Greenland, like you said, I expect like that. The source of phosphorus is already like depleted in the coastal streams and the lake, and that caused a different level of phosphorus limitation into the area. And for the fourth question, the question is, can you?
So production of
Uh-huh.
increase.
Yeah, right.
So these results are a snapshot now.
Mhm.
You think they change?
I think there's a possibility that the anthropogenic source of nitrogen can change the general trend of the nutrient limitation, but I don't think it will like dramatically change the distribution I showed, because that's the like balance between the supply and demand, and like anthropogenic inputs of nitrogen is about the like supply part, and that increase of supply has the potential to increase the demand part too, which again balance the autotrophic uptake length there. And when we see the change of autotrophic uptake length following the river network, we found that it is the short flow is more likely in the small watershed, where light limitation is more likely there. And in that part, like the addition of nutrient may not be a big impact, because productivity is not affected by the nutrient level there, but by the light availability. So I think we have like the addition of nutrient have like very effect in the like small watershed and the large watershed, but in summary, I don't think there will be like very like large change made by that, unless that change is like a humongous amount.
Put item there when you say during summer months, productivity limitations, likely my recollection of the description was that was driven by higher discharge in the summer.
No, low discharge. Mhm.
My suggestion is to think about the data set geographically, typologies of discharge patterns, and not high and low in the same months everywhere. You have maybe hundreds, but this is hundreds of different locations. Maybe you talk to Jaden, you got this map of discharge patterns. You know,
Mhm.
segmenting those by
Mhm.
which phase of the year is high or low discharge.
Okay, yeah, thank you.
There's also a question in the chat for the Arctic streams. Was the higher amount of rain, particularly in coastal areas during last spring and early summer of 2023, a factor of the results?
So for the Arctic streams was the higher amount of rain, particularly. For the comparison between the coastal and inland stream, I did not conduct the experiment comparing the two area in 2023, so I cannot like figure out whether that caused the difference in the nutrient limitation between those two. But that precipitation can be one of the factors, because with the high flow variability, it can cause the scouring of the biomass there, and also the higher light attenuation. So yeah, for sure, that difference in precipitation can be the cause of like difference in nutrient limitation.
Okay, well, thank you again. That's pretty much our time. I believe there's contact information on the flyer for this week. If anybody has any follow-up questions or anything like that, but otherwise I will see everybody again next Monday. Thank you. Thank you so much. That was great.
Thank you.
I really appreciate how like, not everybody kind of is cognizant that there's interdisciplinary.
Yeah,
elements,
right,
and so they view
right.
The presentation, like everybody's in the same field as them and I'm always.
Yeah, I want to make my presentation like as plain as.
Yes,
can,
yeah,
but
which I appreciate
know that.
a lot.
But
Yeah.
there's still like many technological terms.